Hopefully you can see that in full screen now. So as Eleanor said, I've had a long interest in the tumor necrosis factor superfamily and how it regulates T cells. And today I'm going to talk about that and give you a bit of an overview of what I think its role is in the immune response. So just to start at the beginning, for those who might not be immunology experts, T cells are important in protecting us from infection and also in immune surveillance of cancer. And CD8 T cells are important in killing both infected cells and altered cells. And CD4 cells have multiple roles. They can have direct anti-pathogen function through cytokines. They're very important and help for long-lived antibody responses and memory B cells, and they also help the CD8 T cell responses. And so these powerful cells need to really be regulated at every stage of their existence to avoid excessive inflammation and autoimmunity while preserving their important function. So our lab, and today, although we do some cancer work, I decided to focus on our work on viruses and how they teach us about the physiological role of these pathways in the immune system. And our lab studies two different models. We like to study influenza A virus as an important human pathogen with pandemic threats ongoing, and because T cells are very important in any universal vaccine because of their broad cross-reactivity against multiple influenza strains. And it's an example of an acute localized infection. And then the other model we like to study has been a favorite of immunologists for many decades is lymphocytic choriomeningitis virus, LCMV, and particularly the clone 13 variant causes a persistent infection in mice. And because it's a natural rodent pathogen, it's been really informative for understanding the physiological response to infection in its natural host. So, upon initiation of immune response, T cells encounter their antigen from the pathogen in the draining lymph node presented by dendritic cells. Then they undergo massive clonal expansion and go out into the tissues to clear the infection. If successful, most of these T cells die off, leaving behind a small number of memory cells that are nevertheless poised to re-express all their functions rapidly upon exposure to the same pathogen. If, on the other hand, the T cells are unable to fully eliminate the infection, the immune system has to come to an equilibrium with the pathogen so as to avoid excessive damage. And in that case, we know from work from many groups that T cells upregulate inhibitory receptors and produce anti-inflammatory cytokines. And they retain a little bit of residual function to keep the virus in check, but not enough to clear it. So it's in this context that we're really interested in the tumor necrosis factor receptor family that I'll tell you about in a minute. And one of the questions we've had is that we know that the initiation of T cell responses are regulated, and then you get this clonal expansion, but then what controls how long the T cells persist out in the tissues? And that's what I'm going to try to tell you about today. So let's delve into some of the molecules involved. During the initial immune response in the lymph node, T cells recognize antigen presented by dendritic cells, and these dendritic cells have receptors that recognize common features of pathogens and upregulate co-stimulatory molecules, CD80 and 86, which bind to a receptor, CD28. And it's the combination of this signal 1, the antigen-specific signal through the T cell receptor, and signal 2, CD28, that tells the T cell that, yes, there's something going on, start your clonal expansion. And these signals induce things like interleukin-2, a growth factor, metabolic changes that allow the T cells to go through these massive rounds of division, as well as survival signals that allow them to survive through these divisions. And a few years ago, or many years ago now, Matt Mesher coined signal 3 to represent a third important signal during this initial T cell activation, and that is the cytokines present in the initial environment that set the stage for the differentiation of what kind of T cells you get. So that seems nice and simple until you start thinking about all the known molecules between dendritic cells and T cells. And in particular, I'm just highlighting a few here. In red are some of the inhibitory receptors. So every time you activate a T cell, you also need to limit its activation, and inhibitory receptors come up on activated T cells to set thresholds for activation and to control T cells in the tissue. But my focus today is going to be on these molecules in pink, the TNF receptor superfamily, of which there are 30 receptors and 19 ligands. And a subset of these are found on T cells. These four are predominantly studied on T cells and bind to ligands on activated antigen-presenting cells. And they're particularly known for signaling cell survival through NF-kappa B and MAP kinase pathways. So the question is, why do we need these additional signals? Oh, sorry, I'm just going to mention that I'm going to talk about two of these today, GITA and 4-1BB, that we study in our lab. So sort of the questions we've been trying to put in context is, why do we need these additional survival signals when the signal 2 survival signals have already taken place? So when and where do we need these signals, and what's their role? So I'm going to give you two examples from our work that I think shed light on this and lead to our current working model. The first will be on the role of GITA and its ligand in chronic LCMV infection, and the second will be on the role of 4-1BB in memory T cells against influenza. And what I'm going to be telling you about is that we think it's not the initial interaction on dendritic cells where this pathway is important, but an interaction on a subsequent set of cells, the monocyte-derived antigen-presenting cells, that are going to be important for controlling these T cells in the tissues. So the first model we'll start with is LCMV clone 13, and we studied the role of GITA in this pathway by comparing wild type and GITA knockout littermate mice. And this was the work of Derek Cloutier, a former PhD student in the lab. And the first thing we wanted to do was to find out if this really mattered to the control of infection. So he looked at the viral load as well as CD8 T cell responses that are known to control this infection. So shown here is the viral load in the spleen, lung, liver, and kidney, day 8, and the kidney at day 45, which is the last place to clear this infection. And we see that there's about a 1 to 2 log increase in virus if you don't have this molecule. And I won't show you all the data, but the CD8 T cells were decreased. They had higher levels of this inhibitory receptor, PD-1, if they lacked GITA, and they made less cytokines. So it seemed like this was going to be important on the CD8 cells. But then Derek actually got a surprise. He depleted CD4 cells because years ago it had been shown that this makes the response more severe. And although the viral load went up, he completely lost the phenotype of GITA having an effect on viral clearance. And so he did some other experiments to deplete regulatory T cells. I won't go into details, and those still preserved the defect. So this pinpointed in this model, GITA was important on the CD4 cells. So it's actually sometimes very difficult when you're comparing a wild type and a knockout mouse to see the role of a particular molecule or cell in infection if the viral load is much higher, 1 to 2 logs in the knockout. And so you get a lot more inflammation. And how do you know what is a direct effect or an indirect effect? So immunologists like to use bone marrow chimeras, and I'll walk you through this to look at cell intrinsic effects. So the way this is done is you lethally irradiate a mouse, and then you give it back bone marrow to reconstitute the hematopoietic system. And in this case, we're using a mixture of bone marrow from a wild-type mouse or a GITA knockout mouse. And we can distinguish the cells that derive from these by using a congenic marker. The gene for CD45 differs by 1 amino acid that can be detected by antibodies. So we wait 3 months, and after 3 months, we can look at the reconstituted immune system, and you could see that we succeeded in reconstituting a mouse with about half the cells from the wild-type genotype and half from the knockout. And our cells of interest were the T cells. So if we look at the T cells, the total T cells, the CD4 cells, the CD8 cells, or the FOXP3 regulatory cells were equal on the wild type and knockout. So this told us GITA is not involved on T cells in the steady state. It's not required for their accumulation or presence or development. So what happens now if you infect this mouse? So this was from the blood here, so we could go on and infect them. So we infected mice with LCMV, and after 8 days, we looked at various T cells. Here we're doing a re-stimulation with an immunodominant CD4 T cell epitope, GP61 to 80. And here we're looking at interferon gamma producing cells with each dot here a cell. And what you can immediately see is the vast majority of the immune response is coming from the wild type and not the knockout. And this was true if we looked at interferon gamma producing cells, cells making IL-2 and interferon gamma. We also looked at a population of helper cells called T follicular helper cells. These go into the B cell areas of the lymph node to help B cells. And all three of these subsets had a similar threefold defect if they lacked GITR. And so it seemed GITR was really important in all the responding CD4 cells. It had a smaller effect, about twofold, on the regulatory T cells. So it seems like GITR has a general effect on T cells, whether they're regulatory or effector, but they have a bigger effect on those expanding cells that are going to fight the infection, which explains perhaps the net effect on viral control. In contrast, the effects of on CD8 cells must have been more indirect because we saw a little difference between wild type and knockout, a small difference in one epitope, GP33, and none in ENP epitope. So I won't go through the entire paper here, it's been published, but our general conclusion was that GITR on CD4 cells was important for them to accumulate. It affected both helper cells, and we actually saw a corresponding decrease in antibodies without GITR, and it affects CD8 cells indirectly. And when we neutralized IL-2 with an antibody at the beginning of infection, what we found is we not only got less CD4 and CD8 cells, but now we've lost that increased magnitude of T cell numbers with GITR. So it seems that GITR was important to allow the CD4 cells to accumulate and provide help for the CD8 cells. So that was interesting, but we wondered why the CD4 cells were responding and not the CD8 cells. So we wanted to know, well, where's the ligand? We need the ligand for the receptor in order to activate the pathway. So we wondered which cells expressed GITR ligand. And I want to just remind you that in addition to dendritic cells, during an immune response, another kind of antigen-presenting cells comes from the monocyte lineage. And so during an infection, large number of monocytes exit the bone marrow dependent on the chemokine CCR2. And they get recruited to sites of infection and inflammation and go into the tissues where they can differentiate into monocyte-derived dendritic cells and macrophages. So another student in the lab, Frank Chang, decided to explore where GITR ligand is expressed. And so I won't go through the details of the flow cytometry, but he set up a multi-color panel to distinguish dendritic cells, and the classical DC1 and DC2 were known to be important in initiating responses to CD8 and CD4 cells. He looked at some other well-known splenic macrophage and myeloid lineages, and then he looked at the two versions of monocyte-derived dendritic cells, one that were more dendritic cell-like and one that were more macrophage-like. And surprisingly, he found that not the dendritic cells that we think about for initiating responses, but the inflammatory monocyte-derived cells appeared to be the main expressors of this GITR ligand. And you can see the kinetics. It goes up about a day and a half after infection, and eventually LCMV infects all the antigen-presenting cells. And in what I won't show you, it seems to block the interferon signaling, and we lose these signals. For those who are experts in the audience, we were eliminating that these were not dendritic cells because they lacked the classical dendritic cell transcription factor ZBTB46. But interestingly, the kinetics was the same on all the subsets. It's just higher on the monocyte-derived cells. And so we wondered if type 1 interferon induced these molecules because they came up so early in infection, and infection induces interferon. And so we took cells from the peritoneal cavity, and we showed that type 2 interferon did not induce GITR ligand, but interferon beta, shown here in a titration, or interferon alpha 4, both were able to induce this pathway. And we wanted to know if this was relevant in an actual mouse. So we used an antibody to IFNAR1, which is a subunit of the type 1 interferon receptor. We gave this to the mice the day before and the day of infection, and then we looked at all our different splenic dendritic cell subsets at day 2. And again, you can see that you can decrease the induction of this GITR ligand on these inflammatory cells if you block with anti-IFNA. Interestingly, in a work I won't show you, we actually saw 4 members of this TNF family show very similar patterns of expression. So what we found is that dendritic cells, which are the cells that we know from many people's work are important in priming immune responses, although they're high in CD8086 and MHC molecules for priming, they were very low in the TNF family ligands. In contrast, these monocyte-derived antigen-presenting cells, which in our hands had about 10-fold lower MHC, and depending on the timing, they still had CD8086, but what they seemed to be particularly good at was inducing these TNF superfamily ligands by type 1 interferon. And we know from the literature, as well as from experiments we did ourselves, that these were not as nearly as good as dendritic cells as priming immune responses. So what was the role here? So our hypothesis was that perhaps this was a later interaction on already primed T cells. So how could we test that? So again, this is a little bit of a complicated experiment, but I'm going to try to walk you through it. Like I told you before, we like to do mixing experiments. Whoops, I'm going too far. And in this case, instead of a bone marrow chimera, we injected mice with an equal mixture of wild type or knockout T cell receptor transgenic cells. So these T cells all had a receptor for the GP61 to 80 epitope of LCMV. We let them circulate for a day, and then we infect with LCMV. Then we euthanize mice at these time points to ask, when is GITR playing a role in the response? And we can detect these cells with CD45, and they either have GITR or they don't. So the first thing we looked at was whether the cells were getting activated. So we took cells out of the spleen, either immediately or 12 hours after infection. And what you can see is that they upregulate within 12 hours 2 activation markers, CD69 and 41BB, another member of the TNF receptor family. And yet there were no differences between wild type and knockout mice. So this said to us that within 12 hours after infection, the cells that we could then recover from the spleen that were specific for this LCMV GP61 to 80 must have seen antigen because they'd all upregulated these early activation markers. And here I'm showing you mean fluorescence intensity, but depending on the marker, at least 60 to 80% of the cells had upregulated this marker. So what about GITR? Well, we decided to look at 2 of the known signaling intermediates downstream of GITR. One was the phospho NF-kappa B P65 subunit, and one was phosphoribosomal S6. And phospho S6 is an important molecule during cell clonal expansion because it regulates protein translation and cell size. And what you can see here, if we track wild type in red and GITR knockout in black, after the first 12 hours, the T cells seem to show signs of activation. They upregulate these markers, presumably downstream of T cell receptor CD28, which can also regulate these pathways. But only after 24 hours, after GITR ligand first appears on the monocyte-derived cells, do we see that the wild-type cells have a higher signal than the knockout. So this told us 2 things. I think one is that the signaling through GITR takes place with delayed kinetics after the T cells already got activated, we presume by the dendritic cells. And secondly, that GITR is giving you additional signaling over and above that initial activation. So this leads to the model that we now is our working model for how TNF receptor families members play a role on T cells. We believe, based on all the work from the literature, that T cells get primed in the lymph node by classical dendritic cells with signal 1, 2, 3. The signal 3 cytokine, type 1 interferon, not only is important here, but it also is very important on the inflammatory monocytes in inducing this family of TNF superfamily ligands. And then these activated T cells can then find the inflammatory monocyte and get an additional signal. And we've looked a little bit more in depth into the GITR signal, where we did RNA sequencing, and there were about 700 differentially expressed genes, some up, some down. But one of the ones I wanted to point out that we could confirm by flow cytometry was GITR signaling allowed you to induce much more CD25 which is a component of the IL-2 receptor. And as I told you in this model, IL-2 was really important for the CD4 cells to clonally expand, as well as the CD8 cells. And it seems like GITR is allowing the T cells to respond to this signal, and that's just one of the things it does. So it seemed to regulate a number of survival signals, and we think this is important for the T cells to persist a little longer so that they can handle the infection. But I was showing you actually a persistent infection model. What about an acute infection model? So our lab has worked for many years on influenza A virus. And we'd shown a number of years ago that if you block the GITR signaling pathway by knocking out the receptor, or the 4-1BB signaling pathway, in this case, we used the ligand knockout, you had decreased survival to a sublethal dose of influenza A PR8 virus. And other work went on to show that in this model, it was actually CD8 cells that critically required this signal. And I'm going to now show you a little bit more about that and some of our thinking about what happens in the lung. So first of all, in this model, are the ligands also expressed on these inflammatory monocyte-derived cells? And so student William Chu looked in the lung, and here's the classical dendritic cell, and here are the inflammatory monocyte-derived cells with either a control antibody or anti-IFNA added before infection. And here you see that, again, it's monocyte-derived cells that express 4-1BB ligand and GITR ligand in a type 1 interferon-dependent manner compared to classical dendritic cells. So I want to digress a minute now and tell you about tissue-resident memory cells. So memory cells come in both circulating and non-circulating subset. So we have central and effector memory that circulate through blood, lymph node, and in the case of effector memory, also enter tissues, and tissue-resident memory cells. And these cells actually come into the tissues as effector cells and differentiate locally in response to local cues, and they actually persist at the site of infection. And a molecule called CD69 is often used to detect this because it controls tissue residency. And they stay there, and they're poised to release cytokines very rapidly on re-exposure right at the site of reinfection. And we knew from work from Jake Kohlmeyer at Emory that monocytes are actually important for influenza-specific tissue-resident memory to form, and that this requires a local interaction in the lung. And we also know that tissue-resident memory in the lung, in particular, don't last that long compared to, say, the skin. They disappear over time. So really understanding what promotes memory of these cells is really important for long-term vaccination. So our work, and I won't show you all the details, showed, similar to the experiments I showed you with the mixed bone marrow chimeras, that cells that have GITR and 4-1BB are important to accumulate in the lung and form tissue-resident memory. And I'll just give you one little bit of data here. Here's our mixed bone marrow chimera again, and we have wild type and knockout bone marrow. And if you look in the lung tissue at the peak of the response, and we use an actual antibody, a fluorescent antibody, injected just before we euthanize the mice to label the blood cells. So by default, the unlabeled ones, we can detect antigen-specific cells. And what you see is, again, like we saw with GITR, you always have more 4-1BB wild type than knockout cells. And this happens already at the peak of the response and persists into the memory phase. At the early response, this is mainly happening in the lung, and we don't see it in the spleen. But later on, some of the circulating memory cells are also affected, and we see this effect. So I'm hoping I'm not running out of time, but I just wanted to give you a little bit more, data here. And that is whether we could actually improve tissue-resident memory cells and circulating memory cells by giving a super physiological signal through 4-1BB ligand. So here we took a mouse and we infected with a variant of flu, an H3N2, and let them clear the infection and develop memory. And we did that to kind of mimic an adult who was immune to flu getting a vaccine. And then we boosted with an adenovirus that was replication defective, either with the nuclear protein gene from flu or having two genes, NP and 4-1BB ligand, each in a different promoter. And then we actually tracked these mice by looking at T cells in the blood for almost a year. and then we challenge them with a different strain of flu that has different antibody epitopes, H1N1, but still shares the same NP gene. So what we found is here's all our different controls. They mount an immune response to flu measured by the NP specific cells, and it contracts, but it's a little bit higher than the original. You have this memory response. But the addition of this 4-1BB ligand in an intranasal vaccine gave us a massive expansion of these circulating memory cells. And they persisted a very long time, out to the 300 days of the experiment, with a much smaller effect if we only added antigen without the 4-1BB ligand. And when we challenged these mice, remember I told you that the response to flu is protective only a few months, maybe 3 months under these conditions. And now we could wait 9 to 11 months. And the ones that had cleared the infection or only been boosted by NP alone lost weight and succumbed, whereas these ones that had had 4-1BB ligand were protected. And we delved into what T cell response we had. And so one thing we noticed is that the T cells we got, not only were there more of them, but they made a lot of different things. They made interferon gamma, TNF, and IL-2, and they could degranulate when they stimulated them as measured by LAMP1 on the surface. We also found that some of them had higher IL-7 receptor that allows them to become memory. And interestingly, the antigen alone didn't work as well. And when we tried to increase the amount, it actually got worse, sort of like giving signal 1 without signal 2. And if we euthanized some mice at a later time point, we could actually dissect what kind of T cells we had in the lung tissue. And we had a mixture of these CD69 positive tissue resident cells, as well as some circulating cells. Now, in other experiments I won't show you, we tested, did you have to give this intranasally? And yes, you did. If you gave it systemically, it didn't work. And if you tried to give the antigen systemically and co-stimulation in the lung, that didn't work either. It really needed the two signals together locally. So one more piece of data I haven't shown you in the interest of time is, remember those signaling experiments I showed where we allowed wild type and knockout cells to compete and we can detect when the GITA positive is signaling? We found that we could see a little bit of this in the lymph node during flu infection at day 3, but it was a bigger signal in the lung at day 5. And we just showed this for GITA ligand. But by analogy with the other members of the family, our model now is that T cells get activated by dendritic cells in the draining lymph node. They may see some monocytes there. Then they get to the lung, and there they encounter a lot of inflammatory monocytes during, for example, flu infection. And we think this is really important in this harsh environment to let these cells get additional survival signals and respond. And this leads to increased T cells at the peak of infection to clear the virus. And it actually increases our tissue resident memory cells and also the circulating ones. And so why is this important to control T cells at the site of infection? Well, earlier work we'd done, if we used a very mild flu and we knocked out this 4-1BB signaling pathway, it didn't really make a difference. But when we used a flu that took about 10 to 12 days to clear, if we knocked out this pathway, the mice died. And we found that it was really necessary in that context to sustain the T cells long enough to clear the infection. So this leads to our current model then of T cell activation, where signal 1, signal 2, and signal 3 take place on dendritic cells in the draining lymph node and initiate this burst of clonal expansion. Then type 1 interferon induces the TNF superfamily ligands on these inflammatory monocyte-derived cells that are massively induced during infection. And we think this happens both initially in the lymph node to a small extent, but much more importantly in the tissue, and that this is really important in giving you a signal that if the virus is there, there's still interferon, you've got these molecules induced, you will keep the T cells long enough to clear the infection. And both these ligands and the monocytes disappear as the virus is cleared. And so we think this is really important positive checkpoint for T cells in the tissue and needs to be thought about during vaccination. So for influenza virus, where we know for tissue resident memory, you need local delivery of antigen to the respiratory tract, we need to think about whether we're also inducing type 1 interferons and monocyte-derived antigen-presenting cells in our vaccine setting to get this protective response. So I'm going to stop there. I just want to thank all the people. The LCMV work that led to the initial thoughts about Signal 4 came from these guys. The flu work was from postdoc and two students. And I haven't talked about Karen. She's a current student doing RNA-seq on the 4-1BB pathway. And I didn't have a chance to tell you about our tumor models, where we also think monocyte-derived cells are important in the tumor if you treat the tumor with a STING agonist to induce type 1 interferon. And many people have given us mice, and I'd like to also thank the CIHR for funding, as well as EPIC at the University of Toronto. So happy to take questions now. Thank you.
Thank you very much, Tania. That was a great presentation, very comprehensive. Yep, there's a whole bunch of questions here to be answered, so I'm going to try and moderate and put some together in a and get the gist of what people are asking. So at the very end of your talk, you mentioned about vaccination and flu vaccines. So my question to you is, our vaccines are not being delivered intranasally for respiratory tract infections. What do you anticipate would be the outcome if we actually use these vaccines intranasally?
Well, we do have one intranasal flu vaccine. That's the live attenuated one. And there's two problems with that. Of course, for adults, we probably neutralize it with antibodies. And for children, it was thought it was going to be better. But apparently, in practice, it isn't better than the inactivated vaccine. And it may be too attenuated. So there was a really nice paper in Science in 2020, and I wish I could remember the author's name, who delivered STING agonists, which induced type 1 interferon. And it was a really elegant model. They used surfactant labeled nanoparticles so that the alveolar macrophages would take it up. And then the part they got delivered to the airway epithelial cells, and then you got really nice interferon response, and they got very good protection in a mouse model. And so I think, we need to think about that. How do you get the right adjuvant to the right place? I think a lot of people, I think Akiko Iwasaki's been a champion of this, think that we should use a systemic prime to get good circulating cells to protect us, from systemic infection, but that we're going to need an intranasal boost. And so the question is, how do you do that? And I think new adjuvants are going to be really important, really tailored to that environment.
Yeah, thank you. So another question was, does GITR actually directly activate the AKT signaling, the mTOR pathway, or is there crosstalk with other factors? For example, you talk about type 1 interferons. So what evidence do you have that it's a GITR direct activation of AKT and not interferon?
Right. I didn't get into the signaling. So GITR binds TRAF2 and 5, and we know the direct link to NF-kappa B and MAP kinases. That's well worked out. When Derek did his signaling experiments in vitro, we pre-stimulate the T cells, and then we add an antibody to GITR, an agonist, and within 15 minutes, he got AKT and phospho-S6. So we think that's probably direct rather than through an intervening cytokine. But my colleague Byung Kwan, who was the discoverer of 4-1BB, published a very nice paper on 4-1BB signaling, which binds a different set of TRAFs, TRAF1 and 2. And he showed that took 3 hours to get to AKT activation. So I think for 4-1BB signaling, it's probably indirect through cytokines. Through GITR, it seems very short-lived. And OX40 is quite similar to GITR. They both bind TRAF2 and 5. And there, Mick Croft has done some quite detailed work on the link to PI3 kinase AKT and some cooperation with the TCR signaling pathway. So I don't know if that's how GITR works as well, because generally these pathways work. You have to have antigen to get the receptors high. So they're kind of working in the context of T cell receptor signaling at the same time.
Okay, thank you. Another question was whether GITR is expressed on other cells.
Yeah, so a lot of our work focused on looking with these mixed chimeras, so we could look at intrinsic effects. It's on NK cells, and many of these TNF receptors are on activated NK cells. GITR has been very puzzling. It seems there's literature saying it has a negative effect on NK cells, whereas 4-1BB actually has a positive effect on NK cells. They can also be on the antigen-presenting cells. So that's why it can be very hard to dissect the effects of, say, infusing an antibody to stimulate the pathway, and why we like to use these knockouts in specific cell types.
Okay, another question was how resident memory CD4 T cells recognize inflammatory monocyte-derived antigen-presenting cells. Is this recognition solely through the TNF receptor superfamily, or are there other molecules involved?
I should say for the flu model, most of our data is on the CD8 cells. And actually, the work from Kohlmeier was also on the NP response. Work I didn't tell you, the flu NP is really sensitive to the monocytes and this signal. The PA, the polymerase, which is not an abundant epitope because it's not a structural protein, is not so sensitive to monocytes. So we think it depends. You have to have a fair amount of antigen for this pathway to work. So although we haven't formally shown it requires antigen, we know if you take the T cells away from antigen, within a few hours, the receptors like 4-1BB and GITA go down. The CD4 cells in the LCMV model, they definitely directly recognize monocytes. We'd isolated them and shown that. In the flu model, it's interesting. It's more important in CD8 cells, and the CD4 cells only show a role in competition. So I think there's redundancy with OX40 ligand. So it's a bit complicated, but I generally think you need antigen on the monocytes, but you need a prime T cell to recognize less antigen. And in the flu model, we know flu gets into those monocytes, so we think it can directly present to the CD8s. In the LCMV model, it only presents to the CD4 cells because those cells can't cross-present. And LCMV actually blocks phosphostat 1 and GITA ligand induction. So
okay,
complicated.
it is complicated. I'm going to hand off now to Payam, who's got a question which was too lengthy to put in the chat. Go for it.
Really great talk, Tania, and really nice to see you. My question is actually talking about the TNF receptors playing the gateway from the dark side of the response to infection. Particularly, for example, with COVID, you have these patients who suffer from ARDS and really aggressive inflammatory side effects that lead to lung failure eventually. And I think there's really interesting work from Nick Skartsis and KJ Tang showing TNF can actually expand regulatory T cells. Specifically, TNFR2 is known to also upregulate CD25, as you mentioned. So I want you to, if you can, give some comments on that.
I've just been reading up on that because we're writing a big review. So if you look at OX40 and 4-1BB, and if you, for example, put them in a CAR T cell that's meant to be Treg, they shut off Tregs. They inhibit FOXP3. But TNF receptor 2 is a little bit of an outlier. I didn't actually talk about it on the T cells. Years ago, we did a knockout of TNFR2 on OT2. It was really subtle on the affected T cells. It really seems to regulate, TNF receptor 2 seems to regulate Tregs really well. And actually, Yanni Borst has nice data that it regulates their metabolism differently than on effector cells. So I think it's a little bit different than 4-1BB, OX40, GITA, CD70 seem to be T cells, effector T cells win over Tregs and those models. That being said, if you look in the tumor microenvironment, the Tregs often express higher levels of the TNF receptor superfamily, and work's been shown that you really need to deplete them before your agonist antibodies work. So I think it can be context dependent, but they seem to have the same role on Tregs and T effectors. They're survival molecules, except TNFR2, I think, is a little specialized in preferentially affecting Tregs compared to the others.
Thank you.
Thank you. And maybe just the last question, because we've already run out of time, was whether you have any insight into the importance of delivering the 4-1BB ligand signal 4 in young versus old age, and wondering if the pediatric immune system particularly needs an external 4-1BB signal when this ligand expression tends to be lower.
Yeah, we haven't really done any age-dependent work. Most of our mouse work's on, 6-week-old mice, or else they're about 4 months old when we do the bone marrow chimera. So I couldn't answer that directly. I think it'd be difficult with regulatory to put a self molecule into a vaccine given to healthy people. So we think the work on this signal 4 really should inform adjuvants and how to get these signals naturally, and that we should be able to monitor that. And of course, it's very hard in human to monitor tissue resident memory. But actually, during the SARS pandemic, people did look at the bowel, and there is work in human, but it would only be in a research setting. It's a little invasive to do that. So.